So, welcome everyone, and thank you very much for joining us for this webinar on heart rate measurements in fish. My name is Anna, and I am the Sales Director for Star-Oddi's implantable loggers. I will be moderating this session, so I just wanted to take you through some practical issues before we get started on the fun stuff. So the webinar will consist of 4 presentations. Each presentation is going to be around 10 minutes. And after every presentation, we will have about 5 minutes for you guys to ask questions. And hopefully towards the end of it, if everything's running smoothly, we'll have about 10 to 15 minutes of a discussion session towards the end. You've probably noticed that all of you are muted. So to be able to ask questions, you will have to use the Q&A feature, which you will find at the bottom of your screen. So you simply press this Q&A button and there will be a window that pops up where you can type in your question, and then I will be moderating the questions in between the talks. So now that we've gotten that out of the way, I think we can just get started. We're going to start off with Ásgeir Bjarnason. He is Star-Oddi's heart rate expert and also the lead designer behind our heart rate product series. He's a biomedical engineer and has been working with implantable ECG since 2008. He's going to give us a little bit of an overview of the company and the heart rate loggers themselves and how they can be used in fish. So over to you, Ásgeir.
Thank you, Anna. Yeah, so let me get this going here. Share screen. All right. Yeah, so as Anna said, I'm going to give a little bit of a technological overview of the heart rate loggers. And then we'll see Tanya, Zoe and Alpin will give you more of a hands-on experience of how to use them in fish. Happy with the sign up. Some of you might know me or have met me, or we've had some Skype sessions. We were planning to have this workshop in the ICBF in Montpellier this year, but when it got postponed, we figured a webinar would be a good option. So now Star-Oddi is an Icelandic company and founded in 1985. It started with large temperature loggers for deep ocean studies, but in '93 it released its first DST, data storage tag, that measured temperature and depth to study fish. And then in 2003, the first implantable temperature sensor was released. And it's part of a certain catalog of tags that has been developed around that kind of implantable logger. And we do perhaps a few thousand sensor tags this year, each year for implants, and some of it goes to wildlife and fish research, but a lot of it lately is going into virology research due to the coronavirus. Now in 2013, as an extension of that, we released the first heart rate sensor, the micro heart, which was designed for laboratory animals, rat and ferrets, but due to our, let's say, knowledge of the fish domain, it saw some use early on, which has kind of expanded over the years. There are currently 24 people working for the company, and all manufacturing is done in Iceland. A little bit more about it. It operates in 4 markets. There's this underwater environments and subsea gears, which are usually they're used mostly in stationary measurement, fishing gear, autonomous underwater vehicles. Then we have the aquatic animals, mostly fish. This section used to be the core of the company, but today it's maybe more around 10 to 15% of the revenue. Then we have laboratory animals and wildlife and livestock, and then some production control, which is mostly temperature, and then some pressure sensors. And this is just these are these like typical data storage tags that we have. They're ceramic, high pressure tolerance, and we offer, let's say, a few different variables. One of the more unique ones that we offer are heart rate, activity, also salinity, and magnetic field strengths. So the telemetry is simple here. It doesn't apply to the archival tagging. It's for like laboratory rodent animal equipment. And yeah. Now, so like maybe a final slide, a little bit regarding the company, and then to transition ourselves into the heart rate talk. Then this graph shows let's say the development of citation using, so papers pretty much using Star-Oddi devices over the years. And we're kind of reaching about 120 papers a year using our devices, which we kind of try to monitor tightly. But what is maybe more interesting here is that the first journal paper using the heart rate sensor in fish was published in 2017. And since then, there has been, well, let's say, a steady increase, having 5 papers, journal papers out in 2019 using the heart sensors in fish, and this year, so far, 4 published papers. And I've heard about at least a few papers being in submission or even, you know, submitted already and being under review. So this is going to be, let's say, a record year, definitely, in terms of publications using the heart rate loggers in fish. Okay, so it's a product series that is constantly in development. Now, as I mentioned, like we started this out with a small one, so the micro heart, 3.3 grams. This was targeted for lab animals, rats, ferrets. This is kind of part of routine measurement, standardized stuff. So it was really like ideal, let's say, for that. But when we released it already, you know, people always want something, you know, different, and they want, you know, more battery life, more memory, etc. So we released a larger heart rate sensor in 2014. And then it kind of, let's say, I think in 2014 were the first sensors that some fish users, let's say, purchased to pilot the stuff. Now in 2016, we made an even bigger battery life, releasing the centi size logger. So then it's up to 19 grams by then. We also did, let's say, a larger memory overhaul of this milli-size logger. We released an activity sensor in addition to the heart rate in 2017. And then we kind of kept on developing. We did a big overhaul of the milli-size and centi size in 2018. And I'm sure that if you're attending this webinar and you have been a user for a while, I mean, I think that you have interest in this. It might be hard to follow if you're a newcomer. And the main thing about the 2018 features were to be able to record lower heart rates. We call it the version 2 for simplicity reasons. In 2019, last year, we released these milli-heart activity loggers and the milli-sized activity loggers. And then this year, we are planning to release, kind of to complete our catalog series, to release a micro-sized activity logger that's already in, we are already doing some pilot work with that. And then we plan on releasing an updated version of the micro heart with larger memory and an updated version of the centi-heart activity. So all our loggers will kind of have the same, more or less the same capabilities, depending on the size. So battery level will be the most deterministic factor between them. So just, this is a quick one regarding, you know, what's the difference between those first versions that we had and the second version. The main difference is that you were limited to 600 samples or 6 seconds of recording with the version 1. This meant that when you're recording low heart rate in fish, because fish, I think when you compare it to the whole animal domain, fish has, you know, among the lowest heart rates in general, and it would clip, let's say. The minimum that we could record was 10 beats per minute, and even at 20 beats per minute, you already started to lose, let's say, records, because The QRS waveform, which we use to determine the heart rate, could fall in the middle of the recording, and then you would be pretty much limited to 20 beats per minute. But we fixed this by increasing the duration. So now you can record 15-second recording at 100 hertz. This enables you to record down to 4 beats per minute, and you're guaranteed to record everything down to 8 beats per minute. So between 4 and 8 beats per minute, you might start to lose some recording, and you can see it here. This is when we were doing the demonstration with this in Atlantic Cod at 2.7 degrees. You can see that the old one would clip at 10 beats per minute, but the new feature would enable you to record this lower rates. Now, here we have now we can kind of go a little bit into the theory behind the loggers, how they work. So here we have the classic formation of the electrocardiographic waveform. So this is it travels through the mammalian heart, let's say, generated in the sinus node, going through the atrium and the ventricles and creating this P, QRS, and T wave. Now, most of the ECG measurement and processing comes from a mammalian domain, where you have 2 atriums, 2 ventricles, and an average body temperature of 37 degrees under normal conditions. Now, birds, they have similar structure to mammals, but 2 atrium and 2 ventricles. Tissue inside is more smooth, and they can generate higher heart rates. Core body temperature is also higher. So we see here 2 ECG, relatively similar looking. One is measuring 80 beats per minute, the other one 400 beats per minute. And then you have fish, 1 atrium, 1 ventricle, blood coming in low in oxygen through the atrium and the ventricles to the bulbous arteriosus, and it's pumping it to the gills where it's oxygenated, compared to the lungs, which oxygenate the blood in birds and mammals. So we have 30 beats per minute, all very similar signals. And this is kind of represents here that you need to record maybe a different sampling frequency to have a certain duration to catch this, let's say, 3-4 QRS waveform to be able to accurately calculate the heart rate. So these are the loggers, and how do we measure the heart rate? So we have these like electrodes, which are platinum based on top of our, let's say, ceramic housing. And so you record, so it has some kind of like a hardware amplification and filtering where it records a small ECG snippet. It can be 600 samples or 1500 samples, or you can actually like program it to do part 600 samples and part of it 1500 samples. If you know that let's say if you're doing a control study and you know that your fish will kind of go into colder water, you can even like kind of program it inside. But you record this ECG, and then it goes through like a filtering process, which pretty much does a peak detection on the ECG. And it stores in the memory of the logger. It can store some of the raw ECG, about 2,700 for the larger ones and about 200 for the really small one. And then you end up with a number, a timestamp, temperature, and heart rate, and associated quality index. Now, when do we save this ECG and why? So you need to verify the amplitude of the signal. So you need to verify your data that it's actually ECG that you're recording and not just movement noise or something. So you need to have sufficient amplitude to be able to kind of differentiate the QRS waveform from other possible artifacts that are recording in this electrical signal. Now, you need to also understand the noise artifacts in the signal. And this is a whole, this is kind of a whole new discussion, because to cover this, we are planning to do maybe a follow-up webinar in June, where we will pretty much just be covering let's say the noise artifacts and how to process them. So that's more for maybe users that have data and want to kind of know more how to interpret the ECG. And then you want to verify this quality index that I mentioned. So the quality index, that was designed to help the user evaluate heart rate calculation and to enable compression of signal for transmission. And it's graded from 0, which is the best, to 3, which is the worst. So usually you have 0, which should be an ideal ECG, and 3, which is noisy ECG that you should probably remove from the data set. And then 1 and 2 can be arrhythmic, or it can be partially noisy data that might generate errors. And it's easy to kind of see, you record your data, and then you do this kind of histogram analysis of the QI indexes, which is possible in the software, just to kind of see, okay, majority of my measurements are QI 0. It means that my recording data set is decent, and I can continue processing that. All right, so then I want to wrap it up with a couple of slides. There's a lot of different motives to measure heart rates. Heart rate in fish. I mean, personally, I'm no, let's say, fish heart rate expert. I mean, my background comes from medical instrumentation and signal processing. My expertise is more in the processing of ECG signals and then recording ECG signals. But through the years, I've got to learn plenty about recording heart rate in fish. And the different motives is, for example, estimation of metabolic rates in the field, connected to temperature and oxygen supply, relating it to climate change, etc. So temperature and oxygen supply also assumed to be key drivers for mass mortality of salmonids, either in the wild, with warming rivers, or in aquaculture. Now, 2 other big factors, stress and swimming activity, estimation of stress and behavior. So this can be in relation to well-being, to transport, to handling, from grading to mortality in aquaculture, or as an effect of temperature and oxygen supply that has effect on these, let's say, parameters. There's a paper out about recreational angling, predator-prey interaction, fishing gear interaction, exploring the relationship with diving, estimation of feeding rates and digestion. Then, going into things such as disease, infection, effect of parasites, or treatment for those, and spawning, or as life event of the fishes. So, I thought about how to kind of wrap this up. So knowing that we have been doing archival tag for a long time, and I mean, it's known that, let's say, location, vertical movements, among other things, have been recorded for years through archival, acoustic, pop-up data. And these are long-term trends often that give certain window into the behavior and environment, benefiting both fisheries and conservation researchers. But that data is often lacking some physiological measurement, relying on environmental variables. Some rely on blood sampling or laboratory data, and therefore there are plenty of questions yet to be answered and mechanism to understand. And for example, here you have Atlantic bluefin tuna doing some diving. So you have depth, internal and external temperature measured every 2 minutes for 24 days. And then on the right-hand side here, this is actually from the data storage tag from us. It's recorded for 1,400 days. So this is wolffish. And you can see this kind of clear annual cycle where it's going to the spawning grounds, and then it's going to the bottom of the sea and up to the spawning grounds, etc. And it's kind of interesting to be able to, have this years of data, and to maybe be able to, take another look at them with some new, let's say, physiological variable measured. Yeah, so this is from these 2 papers. With that, I'm going to wrap up my talk, and next up is Albin Grans of Swedish University of Agricultural Science. He's going to talk about how to make the most of studies using heart rate loggers in fish. And after that, Tanya Prestai, currently with the Marine Institute in Newfoundland, Canada, is going to talk about measuring heart rate in nesting smallmouth bass and spawning sockeye salmon. So both Albin and Tanya have published a few papers using this logger. And to wrap it up, it's going to be Rose Rinnie of Memorial University of Newfoundland, Canada. She's been working in Kurt Campbell's lab. They have used, she's going to talk about using heart rate and acceleration as well, the tags in Atlantic salmon and lumpfish. And I think with that, we give it over to Albin. Or
Yeah,
Anna, no,
so
it's 5 minutes. Yeah, now it's
yeah, so
questions.
you have 5 more minutes. So there's no questions so far. I should have mentioned earlier that you're more than welcome to send in questions at any time. But
I ran through it. I looked at the time and I saw that I was doing it way too slow, so I had to kind of speed up.
no, but we're making good time. But if there are no more questions, then I think we'll just let Albin go ahead.
Sounds good.
Okay, thank you. Let me share my screen. And there it should be. Okay, thank you for organizing this. It's going to be really interesting. My name is Albin Gräns. I'm in Gothenburg, Sweden. It's getting dark here. I've been working with the implants pretty much since they came out. I think me and Mikael Axelsson, who some of you might know, we ordered a logger early 2014, and I looked it up. It actually has serial number 9, so it must have been at least quite early in the process. And I saw from the title I sent in that it sounded pretty ambitious, and I probably won't, but maybe I'll give you some tips and tricks. I was asked to to mention some of the things that you do, but you can't really explain in the papers maybe. And Asker asked me to go through a little of the surgical and post-operative procedures we use. So over these years, we developed a protocol that works for us. It's constantly in development, of course, but and basically what we see, like sterilizing all the equipment, both surgery and implants, of course, is important. We use Sidex if possible. If we don't have that available, we use ethanol. We anesthetize the fish, usually in MS. We've been working with benzocaine as well. And then if the procedure, any procedure we do is more than 10-15 seconds, we bring the fish up to the surgery table so we can give it oxygen during the procedure, similar to the one on the figure there. And to position the implants, in rainbow trout, what we have done is that we make the wound about 1.5 implant from the pericardium and introduce it, the implant upwards, anchor it above the incision wound. It makes a really big difference if the implant is pushing on the wound or not. And this works really well for cod and trout, whitefish, salmon. We have also, in big salmon, we have turned the implant around. So we actually tunnel it up and introduce, so we put the anchor stitch in this area instead. So we pull it in so we can make the incision a bit further back, which has also been giving good signals. We've been looking into how to rotate the implant, and from the first ones, the anchoring tunnel could pretty much go in any direction, and that's been improved. I can see on the old one I have in my hand, we actually made a new tunnel to tunnel it the way we wanted it. But the best, you can get good signals with whatever rotation, but the best for these guys have been if the 2 electrodes are facing the abdominal wall. So actually the opposite of what is shown in this one and what it's shown here. We also tested the importance of actually keeping it as close as possible to the heart and just taking it a couple of centimeters back. If your incision wound was a bit too far back, it makes enormous difference on the signal amplitude, which is of course not surprising. I have to say that this is the method that works for us and for the species that we've been working with. From experience of working with lungfish and some other species, I'm sure this positioning would not work. And I know colleagues that need to rotate the implant so both electrodes pretty much are almost facing and touching the heart to get good signals. So it is a bit of a trial and error to get it working. So once it's in place, we close the wound with sterile monofilament non-absorbable sutures. We use interrupted stitches. If one comes out, we don't want the whole wound to open, but at the same time, they should not come out if you do them correctly. We usually treat our wounds with 2 different kind of powders, one antibacterial, one antifungal, and we cover it with a protective paste. This is actually something we never tested. We've developed it and it's been working, so we haven't stopped doing it, but I would like to look into how this actually affects the healing process and the wandering back of the mucus after you make the wound. But that's a later study. If you do this in a good way and it works, this is pretty much what the wound looked like. This is after three weeks. And why we put so much time into this, one reason is our goal is to release them back in these cages with thousands of other specifics, and we will never be able to actually check them after the surgery that they're doing all right. So we need to have a protocol so we feel safe that they are doing well. And you can see how the anchor wound is in front of the stitching. So this is after 3 weeks with thousands of other fish in a sea cage in the Baltic. And we don't want it to look like this. So this is another experiment that we had to terminate and rethink and redo. And the interesting thing is these 2 were running in parallel. And the only thing that we could for sure say differed between these two surgeries. I don't know exactly how this one was done because I wasn't attending those surgeries because I was doing these ones. But the only thing that we know for sure, we were using the same protocol, was that in the one that looked good from the Baltic sea cages, we were actually using antibiotics, which we could not in the other one. So lately we've been looking into whether or not to use these broad spectrum antibiotic when we do these surgeries, when we're going to release them in for a long time. And this is some results from one of those, from that study. And first of all, you can see, which is, I guess, a bit troublesome for some experiments, but it takes some time for the fish to recover. And we've seen this in cod, we've seen it in salmon, we've seen it in trout and whitefish. It takes at least 4 to 7 days to recover fully and get the stable new baseline of heart rate after going through one of these procedures. But from this study, so the blue is an untreated group and the black is an antibiotic treated group. So we did have an effect during the first week, but it was actually the opposite of what we expected. We had an increased heart rate with the antibiotic. So what does this mean? Does it mean anything? Well, it was actually also translated into a significant increase in an indicator for an inflammation. I have to say that all these fish actually looked pretty good. They were in good shape and there was not much, but it's still an effect. So, and this was a lab study, but so yeah, we don't really have any evidence that that was actually the cause of what happened earlier. Another interesting thing from this experiment is actually that more effect than the treatment was actually who held the knife during the procedures. So we had a more experienced surgeon and we had a less experienced surgeon, and that effect was larger than the one from the antibiotic, and even more surprising that that effect lasted throughout the entire experiment. So even 3 weeks after, that difference was maintained. And talking to people that do a lot of surgeries, that they're not surprised, but it would be interesting to know why it's like this. So once the implant is in and we start measuring something, we were also interested in, so how accurate is this? So we did that by combining the measurements of the implant with a wireless system that we use. So with the wireless system, we basically get all the heartbeats from a free-swimming fish. And then we can compare that to the Star-Oddi at different intervals, different sampling frequencies, etc. And if you want to read about the details, look at the paper. But so basically, if you have a healthy rainbow trout at 10 degrees sampling for 4 hours, you get something that looks like this. So on the x-axis, we have the RR interval of a beat, and on the y-axis is the time until the next beat. So it's basically the same what the implant is using to calculate the heart rate. But what's important is that, like, when you often talk about an average heart rate, that in this case it's 23, but in reality, the heart is beating all between 60 and, in this case, maybe 12, 13 beats per minute. And you actually, to get an accurate heart rate, you need to catch all these beats. And like Asker was talking about, well, first, obviously, the more samples you get, because that's pretty much random picking these points from the plot, the more you use, the more accurate to read your data will be. But like Asker was saying, that like with the old version, you had a limitation to 100 hertz and measuring for 6 seconds, which basically gave you a cutoff. I know that you could pick up lower, but basically gave you a cutoff around 20 beats per minute. So although you got 70% something of the readings, you will have a biased result, because these will not be representative of the heart rate, at the mean heart rate of 23, but actually a higher heart rate than that. With this new feature of the long one, you can solve this, but we have fish, trout swimming at this temperature that actually would be outside this one as well, and we have to go down to, we have to use the long, but even with the 80 hertz, we don't really use that. I tend to use the long rather than the 80 hertz. One way of getting around that, and Asker was saying some applications for saving the buffer, saving the ECG. But what we've been doing is actually trying to do more with the ECG and saving as much as possible. So in this study on Atlantic salmon, which was held at 4 degrees, we were measuring 8 hours, pretty much, 12 hours of almost constantly ECG saves, to get the really high resolution on what was going on. And what you see is 4 different consecutive stressors that we planned. And from this fish, it's obvious like how they respond with the peak from the stressor that rapidly decrease when we stop. This is lowering the water level, followed by second, a third, and a fourth. And this is what we expected. This is another salmon that went through exactly the same protocol. And this one, well, we had an increase from the first stressor, but then for the following 3 stressors, we actually have a decrease in heart rate. So why is this? And if we didn't know what we actually done to this fish, it would have been quite difficult to interpret this. And I think the answer to that is actually in the heart rate. So now it sounds low, but it's really low temperatures in big fish. So at 30, these are coming to a level where they actually can't recover and they can't respond to it. And we know that the fish to respond with these decreases is actually more prone to get in trouble and that won't recover from a stressor. And we did another study, and this is actually not with the Star-Oddi, but with another implant where we followed a fish for 3 weeks in a sea cage. And what we have on the x-axis is the heart rate of the fish swimming around. And on the y-axis is the response after a burst of activity. And what we can see is that it's more than actually what stressed the fish. Like the effect we get is all, well, largely at least, depending on the heart rate it had when it happened. So what I like to say with this, and that's my final thing, that you really need to know your fish in order to understand the data collect, especially when you do it with fish that are out of sight. And I would say that with trout at 10-15 degrees, we know a lot, so we can interpret it a lot. But every time we start moving into new species, this is a big challenge for us. And of course, a lot of people have been involved in this, collaborators. But that's all I had.
That's great. Thank you, Albin. We have a few questions now. Kurt is asking, what specifically is the protective paste that you use?
It's called, I'll have to double check. I think it's called Aura paste. It's actually, so what's it in English? It's stomipåse. So if you have an opening in your abdominal, you have this like opening in your stomach and to the surrounding, and this is actually the paste you use to seal the bag to your stomach. And I think it's Aura base, I think the name is.
Okay, great. And then Marco had a question about painkillers after surgery. He asks, what about giving the fish some sort of painkiller after surgery? May that help managing the recovery time? Is that something you've looked into?
We not with the implants. We have looked into it with the wireless system. Actually, so the painkiller we saw there was that the fish got a sedative effect of it. So which was not really what we were after. We didn't want. We had fish that had a heart rate of down to like 12 beats per minute in 10 degrees trout. So and we are looking further into that because of course we want to shorten down the recovery time, because that would
of course,
help everything. But so far we've not been really successful.
And we're getting a little behind on the time, so we will take just one more question and then hopefully we can cover the rest in the discussion session. So Amanda asks, do you know if spawning females with loose eggs get similar heart rate readings as other fish using the same implant methods you had mentioned for your rainbow trout?
I don't know. Sorry. I would guess yes, but that's all I can say.
Yeah. Great. Well, thank you so much for that. Like I say, we'll get to the other questions a bit later on.
Yeah.
But next up, we have Tanya, and she's going to tell us a little bit about her work with nesting smallmouth bass and spawning sockeye salmon.
Thank you. Okay, hopefully you can all see that. So yeah, my name is Tanya. I am currently doing my PhD at Memorial University, but I'll be talking about the work that I did during my master's with Steve Cook and Erica Lyson at Carleton University. So this work involved using the heart rate loggers to measure heart rate in smallmouth bass and sockeye salmon. So my experience with Star-Oddi's heart rate loggers have been primarily with using the milli heart rate and temperature loggers. And we were using them to explore the relationship between individual heart rate performance and reproduction behavior in fish. We selected to use the Star-Oddi heart rate loggers because we wanted our fish to be free swimming in the wild, and this was an effective way without having to keep them confined in a cage or tethered. So in this presentation, I'm going to be talking about the different points that we considered when designing a project with these loggers, and specifically their application in the wild. So I'll be looking at how we, first of all, the fact that you need to retrieve the loggers, as mentioned earlier in another presentation. They do not transmit data, they're biologgers, so you do have to collect the loggers in order to get your data. The second one is logger placement. So we were looking at, so I'll be discussing how we place the loggers in different fish according to their different anatomy, as well as how we minimized behavioral consequences of surgical process on the reproductive behavior. So how we modified our different surgical approaches to mitigate the trade-off between using heart rate loggers and monitoring behavior. So to address the first question on logger retrieval, that involved just selecting a right species at the right time. So we focused on looking at smallmouth bass because during parental care, the males tend to nest for 2 to 3 weeks where they clean debris off the eggs or they chase away conspecifics. So we were able to find these nests and capture the smallmouth bass using a rod and reel, and then implement the heart rate loggers. And then we could then release the bass back into the lakes. This is a lake in Ontario, where they could resume their parental care behaviors. We also decided to focus on sockeye salmon because they're simulpara species of fish that die after spawning. So there's a stock that spawn in Gates Creek, which is a man-made channel, which is shallow and approximately 3 meters wide. And so this enabled us to be able to find and retrieve the carcasses and the loggers after they died of natural mortality. So we had 2 implantation techniques according to the location on where the pelvic fins were between the different fish. So in sockeye salmon, we could create an incision that was about 3 to 5 centimeters long between the pectoral fins. And this enabled us to implant the loggers. So we do a vertical approach, which is a little bit different than the approach Alvin was describing. This is where we attach a monofilament to the loggers through the holes that were provided by Star-Oddi on the loggers. We oriented the electrodes facing the pericardium membrane and then guided the logger through the incision hole so it was as close to the pericardium membrane as possible without actually making physical contact because we didn't want to add extra pressure. We then fed the suture through the musculature and then tied an anchoring knot just outside. And then so we could then cut the suture, the monofilament, and then close our incision. And that's pretty much all the surgery. We found this was very effective in sockeye salmon. However, the incision approach in smallmouth bass is a little bit more challenging because they have their pelvic fins more anterior. So it made it more difficult to create an incision so that you could actually place the logger close enough to the pericardium membrane. So to navigate this, we created the incision off center, so more on the side, and it's the same length incision, and the reason it's 5 centimeters long is so you can actually put your finger in and guide it, and guide the logger in place. So as you can see in this picture, the logger is then getting inserted, guided so it doesn't rotate, and then anchored in place. So this method is a little bit invasive in the sense that you do need to guide the logger and get it close to the pericardium membrane. So one thing to be cognizant about is that you do have the liver there, so you don't want to have the logger pressing against the liver. Neither do you want to accidentally press against the pericardium membrane or have the needle of the suture puncture the membrane. So that's one thing to keep in mind if you are using this approach. we found this approach to be very effective and repeatable using in sockeye salmon, Atlantic cod, smallmouth bass, and even in largemouth bass. So for the surgical setup, we also wanted to mitigate the impacts of the surgical process on its effects and its effects it could have on fish behavior, because we were trying to monitor fish behavior as well. So the first way to do that was to mitigate the effects of hypoxia during the surgery process. And this was done by having a tube inserted into the fish's mouth. So these are two different setups that we have. The first one is a tote that recirculates water, where there's a pump in the tote with water that then transfers water over the gills. And in this situation, we have more of a trough that's filled with water, but it also has a tube that's feeding water from the lake or from the channel that's run over and ventilating the gills. To mitigate the side effects of handling time and anesthetics, because studies have found that anesthetics such as MS-222 may actually alter fish behavior and cause a bit more of a drowsy side effect after the surgery that lingers for a while after the surgery. So to manage this, because we wanted our fish to go back into the wild and continue their reproductive behaviors, we wanted to be able to not avoid using MS-222 and having these side effects. So to do this, we used electrosedation fish handling gloves. So this is not actually an anesthetic, it's more of a way of immobilizing the fish. And these gloves function by having this electric box that the wearer attaches on a belt around their waist. Then there's these wires that then attach to the gloves, and these are fabric gloves with a wired mesh weaved into the fabric. And this ultimately runs a current, a minor current, through the user and the fish upon two-hand contact on the fish. You don't feel anything when you're using this, but it does immobilize the fish and enables you to do the surgery. So when you let go of the fish can then resume movement. So we found that once we released the fish, they were actually able to resume their behaviors, and this is actually a very effective way of monitoring heart rate in wild fish that you did want to look at their behavior. So the smallmouth bass resumed their parental care behavior. You see in these pictures, they're tending their nest by removing debris and chasing predators. Similarly, the sockeye salmon seemed to resume what we believe to be normal behavior by schooling with other fish that were not tagged, so you can see the tag in the pink here. As well as exhibiting competitive behavior for a mate. So for the bass, the surgery time from capture, so from when the hook was set, because we caught them with rod and reel, and brought into the boat, and the loggers were implanted and they were released back in the water, was on average 7 minutes. And then the time for the smallmouth bass to resume parental care was just under 6 minutes. So in general, we did tag 22 bass. 3 fish did abandon their nest, so we did not see them actually return to their nest. And 2 fish had logger failure. For the sockeye salmon, we tagged 64 sockeye salmon. 4 fish had logger failures and 5 loggers were lost. We can't retrieve them. I don't have a good surgery time for these because we did a bunch of other processes as well in the surgery, such as attaching Peterson discs. In terms of the heart rate traces themselves, so we gave the sockeye salmon 11 hours before we started recording heart rate, so 11 hours post-surgery. The loggers were programmed to measure heart rate every 5 minutes and record ECG every hour and a half. And we recorded at 100 hertz, so for us it was the older loggers, so the 6 seconds. And this was to optimize our ability to detect lower heart rates. So on the y-axis here we have heart rate, and on the x we have time, and these bands are day and night bands, where the black dots are heart rate and the line is temperature. As you can see, our heart rate appears to have stabilized by the time we started recording heart rate in the field. We don't see a big recovery after the surgery. This is the same case with the smallmouth bass, where they were given 9 hours post-surgery to recover, and they were recaptured 2.5 days later. And heart rate was recorded every 2 minutes with the same hertz as before. And again, they did not appear to have a huge effect after surgery. So to validate our heart rate readings, as mentioned earlier by Ashgir and Alban, recording ECG is really important to both validate that your loggers are recording the heart rate that you actually think they are recording, and that your QI indices, so the loggers will give you a quality index where 0 means a good quality heart rate reading and 3 means poor heart rate quality reading. So you can use these ECG traces to make sure that your heart rate records and your quality index records are trustworthy and you can rely on them. So we are using Pattern Finder software, which is provided by Star-Oddi, and this is an ECG trace provided from the sockeye salmon, where we have 65 beats per minute, and this is 48 beats per minute from a smallmouth bass. Something to note here is that the amplitudes are different between the two species. So on the y-axis here, it ranged between 100 to 1023, which is actually the maximum. And in smallmouth bass, it ranged between 599 from the top, sorry, to 471. So it's much more narrow amplitude. And this is something to be cognizant about when you are deciding on how you're going to place the logger, because Alvin suggested the distance from the heart will affect its signal in the records. So in conclusion, we found that the Star-Oddi heart rate logger and temperature loggers were an effective tool to monitor heart rate in free swimming fish, provided that you can get them back. And the electrosedation gloves can be an effective alternative to an anesthetic if you need fast recovery the way we did, where we can't actually hold the fish off-site for a long period of time to enable recovery, and we needed them to return to normal functioning. And then the logger insertion approach can vary depending on the species, so it's important to know the anatomy of your species and as well as the strength of the signal. So that's it. Thank you.
Thank you, Tanya. That was really great. We have time for a couple of questions. So Sandra Binning is asking, how close to the site of capture did you release the bass after the surgery?
Sorry, how close to what?
How close to the site of capture did you release the bass?
On site. So the bass were actually, the surgery was conducted on the boat, and then we released them perhaps 10 meters, 20 meters from their nest.
Okay, so basically the same place.
Yeah.
And then we have a question from William. He would like to know how you selected 11 hours as your post-surgery recovery time.
So we were trying to minimize the risk of losing our fish, so we wanted to start recording heart rate as soon as possible. So based on literature, there's longer the better. So we kind of just decided that starting at midnight the day of tagging was kind of when we're going to start the loggers and hopefully see that they had recovered. But generally, you do want to have upwards of 10 hours if possible, was from what I got from the literature.
Yeah. We'll take one more. Curtis Pano is asking, how long does the tag placement portion of the surgery procedure take?
That depends on experience, I found. The tying portion is perhaps the bulk. Well, creating the incision is perhaps 1/4 of the procedure, and then the tying would be the bulk of it. So I'd say overall, when you actually have the fish on your surgery table and you're starting, it's probably close to 5 minutes.
Okay. Great. Okay, so I think we'll move on to Zoe now, who is going to tell us about heart rate and acceleration tags in Atlantic salmon and lumpfish. So go ahead.
Okay. Let me get this set up. Okay, thank you, Anna. So I recently finished my MSc program at Memorial University, where I was using Star-Oddi heart rate and acceleration tags to study Atlantic salmon and lumpfish. So I'm going to tell you about a few of the projects that I worked on. In the first project that I worked on, my goal was to determine the reliability and usefulness of data recorded by the Senti heart rate ACT tag. So that's the larger tag that records heart rate and activity. So I recently submitted that paper to Animal Biotelemetry, and hopefully soon a preprint will be available for everyone to read. The first step of that project was to do preliminary trials for tagging. So as Elvin and Tanya alluded to, what I found was that location and orientation mattered. And I found similar results to other papers that have been published so far. The two most important things are placing the tag close to the pericardium and the orientation of the tag. So far it seems that the orientation of the electrodes towards the muscle rather than towards the viscera of the fish seems to provide a better quality ECG. So it's good to look at those two publications to read a little bit about that. And if you're working with a new species, you might want to consider doing something similar. Additionally, I found it useful to use anchoring sutures. I didn't actually use the anchoring kind of opening that's in the tag. I just tied the sutures around the tag itself, just for where I wanted to put the tag. This helps secure the tag close to the pericardium. And additionally, for the acceleration values, it makes sure that the values reported are just due to the movement of the fish and not the movement of the tag inside the body of the fish. So I have posted a link to Google Drive, but I'll also post it in the chat after this. I've posted some of the videos that I have for my surgeries, but in the surgeries I have 4 main steps. The first is to make the incision, which for the salmon is about a 3 to 3.5 centimeter incision. Then insert the tag. You can see here that the sensors are facing the muscles, and I'll insert it towards the tail, and then I'll pull it closer to the pericardium. And then to attach the anchor sutures that I talked about and close the incision with a continuous stitching. So in my first experiment, I was interested in looking at the relationship between the acceleration parameters that are provided by the Star-Oddi software and the swimming speed of salmon. So I looked at external acceleration, which I may refer to as EA, which is when the tag is measuring acceleration above normal gravity. And this is measured in milligravitational force, or millig, and I measured at 1 hertz for 60 seconds. At the time, I chose to average these values over the 1-minute sampling period, but now the software provides, over your sampling period, a minimum, an average, and a maximum EA value, which just makes the calculations easier, but you can also get the raw data as well. And also variation, or VAR, which is the variance in that external acceleration. And to calibrate these values, I performed a critical swim speed test on the fish. And what I found was that EA in panel A here and VAR in panel C increased exponentially with swimming speed. Additionally, I took videos of the Euclid experiments, and I calculated the tailbeat frequency of the fish, and I found that the same relationships occurred with tailbeat frequency. What I found was that variation has a pretty steep exponential equation with swimming speed and tailbeat frequency. And I found that these values in the videos that I took and in my behavioral note-taking, that they were related to the burst and coast swimming of salmon. So in future tank experiments, I could use EA to indicate the swimming speed and tailbeat frequency of the fish, and I used VAR to indicate the amount of burst and coast swimming of the fish as well. So as I mentioned, I did some tank experiments where I was mostly focused on the recovery of the fish. So I held them for 1 week at 11 degrees and 6 weeks, where one trial was conducted at 8 degrees and the other one had some fluctuating temperatures. And for the sake of this presentation, I just want to briefly tell you about the results of the heart rate recovery in these fish. So again, I recorded all of the different parameters over 7 days and 42 days in 2 different trials where the temperature remained stable and the temperature fluctuated a little bit. And like many other researchers, I found that the heart rate of salmon recovered over the first 3 to 4 days, and I determined that using linear mixed effects models. But when I look at the stable temperature experiment and longer holding, using segmented regression and linear mixed effects models, I found that the heart rate actually took between 2 and 3 weeks to recover. So as Elvin may have mentioned, this could be related to anesthetic use and potentially related to inflammation, but it just goes to show that the recovery is much longer than we previously believed when you give them longer to recover. So in all of my experiments, I found that survival of all the salmon was 100% and that the tags remained quite close to the pericardium. And by 6 weeks post-surgical recovery, I found, well, only 10% of the incision sutures remained intact, 80% of the incisions had fully healed, And those anchoring sutures that I had put in, 70 to 80% of them remained intact as well. However, we recently did a field experiment at sea cages in Newfoundland, and I found that there was some premature mortality and tag loss. So for my tank experiments, I've been using non-absorbable silk sutures. So I recommend it may be a good idea to use a different type of non-absorbable suture like monofilament, and consider, rather than doing continuous stitching and interrupting stitches, if you're looking at doing a longer tag experiment. So something that many people are interested in is the quality index values. Just as a reminder, it kind of indicates the general quality of the ECGs that are being recorded. So I looked at the difference between, so I looked at the absolute difference in heart rate between what was calculated by the Mercury software and when I manually calculated heart rate. And I found that the software can miscalculate heart rate when QI is greater than 0. So based on this, we recommend, of course, saving ECGs whenever possible. Of course, when you're first validating your data, you want to, of course, save as many ECGs as possible. But even when you're doing longer-term experiments, you may want to sacrifice sampling frequency for saving ECGs, or saving ECGs a couple times a day just to make sure the quality is still good. And not only calculating heart rate when QI is greater than 3, but also when heart rate is outside the typical range of the tag species, because that can include even when QI is equal to 0. So over all of my experiments with salmon, I found that the majority of my recordings were good quality, and using the method where I calculated heart rate when QI is greater than 0 and when it's outside the range of the species, I found that I lost very little data, so that was really nice. And there is one exception. You can see here about 5% in one of my experiments were QI equals 3 values, and this indicated when there was only one or no R-to-R interval. And that was because in 6 weeks holding at 8 degrees, the heart rate was so low that with the 6-second recording, it could only pick up one or no R-to-R intervals, as Theo mentioned at the beginning of the talk. So now they've come out with a long ECG option where you can record up to 15 seconds. So that should allow research to be conducted at cold temperatures and in species with lower resting heart rates. So I also used the micro heart rate tags. That's the smallest heart rate tag in lumpfish to study their heart rate response to pressure, temperature, and oxygen. And it's worth noting these experiments, we took the fish down to 800 meters in a hydrostatic pressure chamber, and the tag still ended up working. So if you are interested in doing research like that, just know that it is possible. So again, I have the surgical videos available online. It's the same basic steps, so I'm just going to go over the differences between salmon and lumpfish. Lumpfish have a thick sucker that lies right over top of their pericardium. So I had to make the incision just posterior to the sucker. And I found that their liver also tends to sit kind of in front of the pericardium. So I sometimes move the incision from the midline a little bit left just to avoid the liver, sorry. And I only use one anchoring suture. And I'd just like to briefly discuss the differences between using the tags in salmon and lumpfish. So the take home here is that the quality of the ECGs and the amplitude of your R wave, as Tanya mentioned as well, it's going to be species specific, and this is likely due to differences in anatomy. So for example, the salmon, when using the centi heart rate tags, had an R wave that was about 500 millivolts, and when I used the milli heart rate tag in the salmon, I saw the same thing as Tanya, where it was all 1000 millivolts, and it took up the whole bin ECG. Whereas in lumpfish, they do have a smaller amplitude, and I think that that's just related to the anatomy of the fish. So the heart lies deeper in the lumpfish's body cavity, and the liver does kind of sit in the way. So when working with fish that have a smaller amplitude, Star-Oddi can work with you to help change the algorithm so that you can still get good heart rate recordings. However, I didn't do that, so it did lead to a few problems that I can highlight here that might happen when you have small ECGs. So, for example, in the salmon, the percentage of good quality ECGs did not change significantly with swimming speed. But in the lumpfish, when I did an experiment where their activity increased, I found that the quality of the ECGs decreased. Additionally, when I did a CT max experiment where heart rate increased with temperature, I also found that the quality of the ECGs decreased significantly, to the point where only 0 to 10% of them were considered good at the higher temperatures. All of that being said, I was still able to manually calculate enough data to still produce results. So I'd just like to summarize the lessons that I've learned in my experience. First, use sutures to anchor the tag in place. Secondly, test the orientation of the tag. So far, it seems that electrodes facing the muscle in a parallel position is the best method for salmon, lumpfish, trout, and bream. That being said, surgical methods and ECG quality and amplitude will be species specific. I want to make sure that everyone allows significant recovery time in order for heart rate to reach baseline values. And when you're looking to calibrate acceleration values with swimming speed, it might be important to take continuous video recordings and behavioral information, as this might be affecting those relationships. I think it's a good idea to save ECGs as much as possible and manually calculate heart rate for measurements of all QI values, not just greater than 0, and this may be able to increase your data usage. And all of that being said, even if you do these rigorous validation tests, it might not always be representative in the lab of what happens in the field. So you may want to account for reduced survival and tag retention in the field relative to in the lab. So I have lots of people to thank, of course, for all of my research during my MSc, and I'd be happy to answer any questions now.
Thank you, Zoe. It's great to get some activity data in there as well. Very interesting. So Curtis has a question about the sutures. I think you addressed some of this, but he's asking why you use the continuous suture and why did you feel like there was an issue using silk sutures versus the monofilament? Yeah, so for all of my experiments, I chose to use silk sutures, not for any particular reason, but it might be possible, given some other information that I've heard about, that might be a better use for longer term studies. I don't know for sure. I haven't tested monofilament, so maybe someone else could speak to that. And as for the continuous stitches, we originally thought the continuous stitches were a good idea. It helps keep everything together, and of course, the silk in seawater over time is going to degrade. But by that point, the incision had healed. But based on what happened at the sea cages, it does seem like maybe an interrupted stitch might be better, just to make sure that each one of them stays in place. There just might be more chance for infection and worse wound healing at the sea cage, just given the amount of, like, the density of fish. So those are my recommendations. Great. And then we have a question from Carrie-Anne. She's asking if you performed preliminary trials for the positioning of the loggers in the lumpfish. No, so I didn't do that in the lumpfish. For the salmon, I had done some preliminary trials, and we had received some information from Alvin that helped kind of choose how we were going to position it in the salmon. But when it came to the lumpfish, I just chose to use the same method. And I found that, so they were still parallel, and the sensors were facing the muscle, or the body wall, I should say, for lumpfish. And so I did find that to be effective, but I think that Alvin mentioned that they might use a vertical method in the lumpfish or some other method, and I'd be interested to hear if that did get better amplitude. That would be worth knowing. Yeah. Okay, great. I think that's it for now.